Hello everyone. Today we are going to talk about a very interesting topic, the role of artificial intelligence in the field of pharmacokinetics or PK. I am Ritu Gupta and I am thrilled to explore this fascinating intersection of AI and pharmacokinetics with you. So let's dive deeper into how AI is shaping the future of pharmacokinetics. So pharmacokinetics, as we all know, it describes the movement of drug through the body in four fundamental stages: ADME, absorption, distribution, metabolism, and excretion. Absorption tells us how the drug enters body. Distribution shows where it is going in the body, whereas metabolism tells us how it will change before leaving the body through excretion. So now, what is artificial intelligence? So it is the field where computers mimic human intelligence. So now some basic terminologies. Artificial intelligence or AI, this is the broader concept of creating machines that can mimic human intelligence. So think about how our voice assistants like Siri or Alexa, they, they answer our basic questions. Now, machine learning or ML, it is a subset of AI, and it refers to set of techniques that are used to train AI algorithms so that they get better in performing a given task day by day. For example, an email spam filter. These filters improve at catching unwanted emails as users label some unwanted emails as spam emails. So moving further, we have neural networks or NN models, which are like a human brain with several layers of neurons. These models excel at performing complex tasks like recognizing face for security features or converting speech to text. So, as we all know, this artificial intelligence is transforming the field of pharmacokinetics with much faster and smarter data analysis. It enables more accurate prediction of drug behavior in the body, speeding up drug development and cutting the cost and time to patients. So now in this slide, let's try to understand these basic AI terminologies discussed on previous slide in the context of pharmacokinetics. For example, we may have virtual PK expert that reviews a drug's ADME data and suggests dose adjustment for different patient populations. Here, AI will behave like a human pharmacokinetist making drug prescribing decisions. Then comes an ML bot or machine learning bot that will learn pattern from the historical PK dataset and will predict PK parameters like clearance or half-life for a new compound based on its physicochemical properties. Finally, these deep learning neural network models, They may help in predicting plasma concentration over time, considering various factors like molecular structure and patient differences. Now, some real world examples that will illustrate the application of AI-based advanced pharmacokinetic models in drug development. So first we will talk about physiologically based pharmacokinetics or PBPK translational method to better understand pediatric drug development with a case study of monoclonal antibody bevacizumab or BEV. Then we would have Roche's approach to determine FIH first in human dose for its novel compound R1350. And next case study will demonstrate how PKPD modeling can guide go-no-go decisions, taking an example of Pfizer's novel compound GP120 inhibitor. This is our first case study, PBPK-based pediatric translational method for monoclonal antibody bevacizumab. So this case study is an excellent foundation to understand how PBPK models can enhance the translation of pharmacokinetic data from adults to children, ensuring safe and more effective dosing. The details of this study can be accessed using this link. Now, the aim of this very first case study was to evaluate the ability of PBPK models in predicting pediatric exposure for monoclonal antibody bevacizumab. This is the backdrop. Pediatric doses are typically scaled from adult doses based on body weight or surface area, called as allometric method. However, these allometric methods often overlook unique physiological characteristics of children and result into inaccurate dose predictions. Now, the strategy for this study revolves around leveraging PBPK modeling using GastroPlus. So, this is fascinating to learn how PBPK models can bridge the gap between adult and pediatric predictions just by accurately considering physiological differences between adults and children. In the upcoming slides, we will explore step-by-step methodology for method development and validation. The process involved 3 steps. First, developing PBPK models in healthy adults utilizing clinical data, and then external validation was done using 5 different clinical studies. Study demographics such as age, weight, BMI, sex, and disease state were matched. Initial simulations were done using GastroPlus default settings, and then 3 drug-specific parameters: vascular, lymphatic, reflection coefficient, and degradation rate constant of unbound endosomal exogenous monoclonal antibody KDAT were optimized. Target-mediated drug disposition TMDD was integrated to capture monoclonal antibodies nonlinear clearance. Model performance was accepted when simulated concentration time profile overlaid observed clinical data. Then, in step 2, validated adult PBPK model was calibrated for disease impact. VEGFA expression levels were increased by two-folds to reflect the higher circulatory concentrations found in patients. Once the model was validated using adult patients, pediatric predictions were made by fixing drug-specific parameters and scaling only the physiological parameters such as tissue size, composition, perfusion rate, lymph flow, and antigen concentrations to match the pediatric physiology. Peer physiology or population estimate for age-related physiology is a mechanistic framework used in PBPK modeling to predict drug behavior in specific populations such as children. So this approach defines 13 tissue compartments shown here in blue boxes, and each tissue compartment has its own volume, blood perfusion rate, enzyme and transporter expression level, and lipid protein volume fractions. All major organs, they are anatomically connected with plasma flow represented by black solid arrows, and red dash arrows, they represent the lymph flow. So lymph nodes will collect the lymph from all organs and will return it to systemic circulation. Mechanistically, each organ is divided into vascular space, endosomal space, and interstitial space. And then this endosomal compartment is further subdivided to account for endosomal processes such as FcRn recycling and target-mediated drug disposition, which is particularly relevant for monoclonal antibodies. So on this figure, the circles represent plasma concentration, and these blue lines are showing simulated prediction for healthy subjects and patients. On this next figure, we see the pediatric patient simulation performed for a 13-year-old male patient weighing 53.65 kilogram using two different doses, 5 and 15 mg per kg, administered intravenously. So this approach helped us understand how This monoclonal antibody behaves differently in children and adults, providing crucial insights for pediatric dosing. Let's talk about the key takeaways from this PBPK-based pediatric translational method. By accounting for adult and children's physiological differences, the spear physiology improves pediatric dose prediction beyond the traditional allometric scaling that particularly relies on either body weight or surface area. This approach can ensure safer and more effective dosing in children, which is critical for both drug developers and regulators. Now, determining first in human or FIH dose is pivotal for setting up the stage for a safe and effective transition from preclinical to clinical studies. In the second case study, we will explore how Roche has navigated the complexities of early stage drug development for its CNS drug R1315. Primary goal here was to determine optimal FIH doses and formulation strategy before entering phase 1 clinical trials. In the background, R1315 is a BCS class 2 compound characterized by low solubility and high permeability. And these properties were expected to significantly influence the oral bioavailability. In the strategy, biopharmaceutical assessment through GastroPlus involved simulation of drug absorption using physiologically based absorption model known as ACAT model. Sensitivity analysis was done to understand the effect of particle size and solubility on oral bioavailability. And then, as a proof of concept, in the second step, different formulations were tested in a 2-cube factorial dog study assessing three different factors: dosage forms, food effect, and dose strength. In the first step, GastroPlus was used to simulate drug absorption using ACAT, Advanced Compartmental Absorption in Transit, a physiologically based absorption model. This ACAT is represented by 9 GI compartments. Represented here by stomach, duodenum, jejunum is split into 2 compartments, ileum is split into 3 compartments, cecum, and then ascending colon. So each of these 9 compartments are defined by unique pH values and transit time. So drug solubility in the simulated physiological media and permeability were used as input to predict the oral absorption across all these 9 GI compartments. So GastroPlus uses differential equation to describe the amount of drug that is released and dissolved, and thus predicting the amount of drug absorbed for these 9 compartments. So simulation results show that only part of the drug was dissolved in stomach, but most of the absorption occurs during gastrointestinal transit. Because the drug precipitated in the stomach gets enough time to dissolve for complete absorption. Hence, drug solubility has minimal impact on overall absorption or bioavailability. Now, parameter sensitivity analysis showed that bioavailability was barely affected by particle size or solubility at pH 6.5. Next, translational study was conducted using dog model, a 2-cube screening factorial design evaluated 3 factors: formulation, dose level, and food conditions. Both micronized drug in capsules, that is solid dosage form, and surfactant solution, that is liquid dosage form, both formulations were tested under fasted and fed conditions at 2 different dose levels. And here these figures are showing plasma concentration time profiles from dogs for the surfactant solutions and for the capsule administration. So interestingly, this statistical evaluation with insignificant p-values shown in brackets, they confirm that there was no significant advantage of sophisticated surfactant solution over the standard capsule formulation. Now, the conclusion of this study design, both in silico data from GastroPlus ACAT model and in vivo data from 2-cube factorial dog study. So they confirmed that sophisticated liquid formulation was unnecessary, supporting the use of simple capsule formulation for phase 1 study. So this approach not only saved cost, but also accelerated Roche's drug development timeline. So it was a great example to show how a simpler approach can yield effective outcomes, saving a lot of time and money. So now coming to the third case study, this study explores the role of PKPD modeling in making critical go-no-go decisions, focusing on Pfizer's novel compound PF 00821-385, that is a glycoprotein G120 inhibitor a crucial outer protein on the surface of HIV viruses. So this study exemplifies the importance of integrating robust PK/PD models in early stage drug development, setting the foundation for informed decision making. So the goal of this third study was to characterize population pharmacokinetics or POP-PK of compound PF 00821-385 in 24 healthy subjects, and then to predict oral dose required to achieve a 1.5 log viral load reduction over 10 days, supporting critical go no go decision. Now in the background, PF 00821385 is a novel HIV-1 GP120 inhibitor targeting CXCR4 and CCR5A clad virus. So in its strategy, first, single ascending dose study was conducted in 24 healthy males and two compartment PK model with first order absorption, well fitted plasma concentration time data using non-MEM software. Then inhibitory Emax PD model was used to describe viral infectivity inhibition using in vitro IC90 data. Then previously published preclinical PK/PD disease model was adapted, first using preclinical data and then human PK data to simulate the clinical doses. Single ascending dose of compound PF 00821-385 ranging from 3 milligram to 1300 milligram were dosed orally to 24 healthy male volunteers with a maximum tolerated dose of 1300 milligram in this first in human study. And 969 plasma concentrations shown here in open circles. So they were collected and population PK parameters were estimated using non-MEM software. Two compartment model with first order absorption, well fitted plasma data, and this compound demonstrated rapid absorption and clearance with absorption rate constant of 0.598 and short initial half-life of 0.3 hours. An inhibitory Emax PD model described viral infectivity inhibition using in vitro IC90 data. A previously published PK/PD disease model that was earlier developed to describe viral, viral load changes for CCR5 antagonist was adapted using only scaled preclinical data, that is animal data and in vitro IC90 data. And then this updated model was used to simulate possible clinical doses of the compound PF00821-385. In the next step, new simulations were performed with the model updated with human PK data this time. Trial Simulator 2.2 was used to predict viral load time profile under once daily QD and twice daily BID dosing across a dose range of 50 to 1300 milligram using minimum, mean, and maximum PK parameters estimated on previous slide. Due to high variability in virologic assay sensitivity, two IC50 targets were used, low IC50 target of 121 and high IC50 target of 489 nanogram per mil, representing the median and upper limit of sensitivity of IC90 value for compound PF 00821-285. So for the low IC50 value of 121 nanogram per mil, 319 milligram of BID dose was necessary to achieve 1.5 log drop in viral load. However, for the high IC50 target of 489 nanogram per mil, the required dose exceeded 1300 milligram to achieve a 1.5 log drop in viral load. So this was even after exploring the controlled release formulation strategy. So here shown in this figure, additional simulations were performed assuming different Ka values for both IC50 targets, low IC50 target of 121, high IC50 target of 489, and different Ka values were tested for both QD and BID doses. So the results suggested that a dose of about 1000 milligram was required for high IC50 target to meet the requirement of 1.5 log reduction in viral load. So these simulations are critical to show the challenges in achieving a therapeutically effective dose. Now, in conclusion, the dose predicted to effectively target 70% of virus isolates was found to be close or above the maximum tolerated dose of compound, which presented a significant safety concern. So the PKPD modeling indicated that effective dose required was too high, rendering the therapeutic window unsafe. So this essentially means that safety margin between the effective dose and safe dose is too narrow for this to be practical. Further, a control release formulation showed some promises, but offering only limited benefit across a narrow range of viral sensitivities. So, in conclusion, the unfavorable therapeutic window and limited application have led to stop the further development of this compound PF00821385. Now, a glimpse of some other applications of AI making groundbreaking impact in pharma industry. So we would take two examples here. Millipore Sigma's Edison's platform. This is a commercial platform converting virtual design, molecular designs, to real world manufacturability. And then Janssen's Prezista, an anti-HIV drug. This exemplifies the shift from batch to smart AI-driven continuous manufacturing for greater pharmaceutical efficiency and quality. So Millipore Sigma Edison software, this is a commercial platform that empowers medicinal chemists by streamlining entire drug discovery process. From designing to optimize, screen, and plan synthesis of new molecules in one single platform, integrating generative AI and CADD. So virtual screening and de novo synthesis in Edison analyzes more than 60 billion chemical targets to identify potential drug-like candidates utilizing generative AI models such as Reinvent to generate optimized small molecule libraries with superior ADME properties. So this platform seamlessly connects with Cynthia. So this Cynthia is a retrosynthesis software which assesses synthetic accessibility scores and proposes viable manufacturing routes for these novel compounds. Now comes Jensen's drug Prezista. So Jensen transitioned from traditional batch manufacturing to continuous manufacturing of this compound, Prezista or darunavir, using AI and machine learning models, using real-time sensor data. So this real-time data is used to detect the manufacturing anomalies and auto-tune the process parameters, thus reducing the defects and keeping pharmaceutical output stable and compliant with both US Food and Drug Administration FDA and European Medicine Agency EMA expectations. So this enabled automatic adjustment to production parameters, thus reducing the pharmaceutical inefficiencies and improving the overall process control. So as we wrap up today, I would like to sincerely thank you for your kind attention. And it's been a pleasure sharing insights with you. And please feel free to reach out to me for any questions. And I would look forward to your questions and any thoughts you would like to share. Thank you.